And then we have the interlaboratory workflow. So this is in trial implementation when a laboratory needs to order a test from a different laboratory. So in this case, the provider orders 3 tests. The laboratory can only perform the first 2 and subcontracts out the 3rd one to another laboratory. So the interlaboratory workflow profile covers this piece. It's similar to, sending orders out and getting results back, but it's different actors. So we're using a slightly different profile here. It's again 251 message, but LOINC recommended, and you can support it. All right, and with that, I'm going to turn it over to Raj.
I am going to begin by speaking about the anatomic pathology workflow, which is an integration profile that establishes the continuity and integrity of the basic pathology examination with the associated data and images that are exchanged between the systems of various care units or ordering units and those of the anatomic pathology laboratory, and then the back-end systems that are producing, archive, communicating, or presenting various supportive data or images for anatomic pathology. The specialty of anatomic pathology is currently undergoing a transformation. I would say most facilities in the world today still leverage a microscope and glass slides, which look at tissue that's 5 microns thick and is viewed through optics in real time to render diagnoses. However, the future is a digital one in which those glass slides have been captured through those optics but into a computer system, and then the pathologist or the interpreter of those images is actually sitting at a computer station looking at the image on a monitor. There are certainly some European countries that are much more advanced than the rest of the world in this. To name one would be Sweden, who has been one of the leaders. However, most countries and laboratories around the world, the United States included, still base most of their anatomic pathology work using a microscope, glass slides, and tissue that's embedded on that glass slide. However, we need to start thinking about the old workflow and the new workflow. Obviously, the new workflow, utilizing digital images, has an opportunity for automation, has an opportunity for interaction of vended systems in a way that facilitates activities that enhance patient care, enhance productivity, that simply would not be possible before, and understanding exactly what needs to be machine understandable and what needs to be human readable and communicated through these various platforms is very important. So next slide. Another part of anatomic pathology reporting is the connection to public health and the transmission of microbiology results, reportable conditions, as well as tissue for specific conditions like cancer to cancer registries. It would be based on the anatomic pathology messaging, but also supporting additional elements that are of interest to public health agencies. This has been Connectathon tested utilizing standards HL7 2.5.1, LOINC, DICOM, instrument CT, using the transaction PAT10, public health reporting, to go from the sender to the receiver. There's a depiction there of kind of that part of the process. There's many situations with an example here given of the CDC and Cancer Registry from the United States of potentially sending data from an anatomic pathology laboratory to directly through automated systems to these receiving agencies that need to get it. Today, I will say in our glass slide and microscope-based mechanisms at most hospitals, might be utilizing electronic health records and lab information systems, but the transfer of information is still contains many manual steps. And the hope is with profiles developing, such as ARPH, that much of this work can be automated and less of it, there needs to be less human intervention for this moving forward. Next slide. So the laboratory specimen handoff profile, again, is speaking about an interchange, but this time of specimens, and passing of specimens between laboratory automation systems and specimen processing devices, which is an integral part of clinical laboratory operation. Current vendors of various devices today have very little guidance on how to implement specimen handoff. HL7 and CLSI Auto 3A do provide some direction, but not specific guidance on this interaction for individual handoff workflows. And in the absence, of course, of any guidance, much of the specimen handoffs are implemented by various vended systems on an ad hoc basis, which means that every implementation at every site needs to implement a custom interaction. And so there is a clear need for a fully specified interface to reduce the time and cost associated with managing these specimen handoffs in clinical laboratories today. And it's our hope that this laboratory specimen handoff profile, which is in development, will provide the standardized workflows for the most common use cases. And you see some depictions of what's involved there. It's important to note that the profiles that speak to automation don't necessarily speak to the handoff of specimens once it leaves that device. So this is specifically focusing on the transactions between devices where it's moving perhaps from one analyzer to another analyzer, and that handoff needs to be managed smoothly and electronically without human intervention if possible. Next slide. So more globally, beyond just the automation, specimen event tracking is important. The current PaLM technical framework does not cover the transactions and workflows to trace all of the events regarding the chain of custody for a biologic specimen, which means it's impossible to completely reconstruct the history of a specimen in the absence of this new profile that's under development called specimen event tracking. And this includes everything in the life cycle of a specimen from the point at which it's collected at the bedside, sent to the laboratory, received by the laboratory, rejected, or stored into a biorepository potentially, or an archiving system of some kind later to be recalled. It focuses both on the transport of a specimen within a laboratory, as well as use cases for transfer between laboratories. And you see a number of use cases listed there, including, perhaps within a biobank or across biobanks, and does include the derivation of samples in a parent-child hierarchy. So if you have a tissue specimen that's embedded into a paraffin block and then you derive slides from it, that would be a child specimen. If you have a sample of blood and there's DNA aliquots that are generated, or RNA, those would all be child specimens, and that entire tracking of where a specimen originated would be part of this specimen event profile. And obviously interaction would need to interact with a number of other profiles, such as the label profile, LBL, the LTW profile, the point of care profile, as well as the automation profiles, LDA and LAW. There's also other profiles in development in terms of anatomic pathology and the digital workflows, which I'm going to speak a little bit more about in the coming slides, and this will touch really all of them, as this touches the specimen, and specimens are part of many other profiles within IHE. So we can go to the next slide. So one of the challenges in the anatomic pathology world has been in the codification of discrete data elements to enhance machine understanding and automation. Most pathology reports of the past, I would say mostly distant past, but also today, consists of verbose text that requires significant natural language processing capabilities in order for a machine to try to deduce what is being written and conveyed in particular aspects of an anatomic pathology report. The APSR is now at the 2.0 version. The APSR 1.0 version, real quickly from a historical standpoint, was an attempt to look at the major common cancers and create all the discrete data elements that were important for capturing those. It was quickly realized that having IHE support a profile that for a field that's always changing, constantly evolving, and would require a significant amount of maintenance and curation. And it was felt that creating a template for a more generalizable anatomic pathology report was probably more pragmatic, and that led to the creation of the APSR 2.0, which you see depicted here, which really conveys the common components of a surgical pathology report. Surgical pathology referencing generally tissue being taken from a patient and then sent to a pathology laboratory, and then a diagnosis being rendered. So this covers surgical pathology and cytopathology. Worldwide, these are the general sections or types of data that are collected, and the profile provides that template for building specific templates for covering various diseases. And the hope is that vended systems might implement a profile like APSR 2.0 and allow the end users to create content which could then be shared because of the nature of the profile. Next slide. This is being authored in Art Decor, where every one of these data elements is being defined and documented. There's a live hyperlink within the presentation, if you have access to it, that will take you to the site with more information. Next slide. There is also a Wiki site that really explains this profile in detail. I think it's very important to have the value of the template for sharing standardized content for anatomic pathology recognized by vendors worldwide, and so I'm hopeful that we'll be able to spread the word and get the vendors that are producing systems that manage anatomic pathology content across the world in various countries very interested in this profile and support the profile so that the content can continue to grow and be shared, be machine processable, and have greater value than the current state in which anatomic pathology data is stored today in vended systems. Next slide. So I wanted to come back to the anatomic pathology workflow briefly to provide just a little bit of history because there's a shift, as I mentioned, towards digital pathology and specifically being able to capture using what's called whole slide digital imaging or whole slide microscopy imaging in the anatomic pathology workflow. And so what's been done is to leverage the transactions that have been documented in the PaLM technical framework, specifically the transactions Lab 1 through Lab 5, and kind of overlay them onto the work and effort that's gone into development of transactions in the radiology domain. So if we go to the next slide there. We see that on the radiology domain, there's significant work already in terms of their management of digital images with an image manager, archiver, some mechanism of acquiring those images, and then, of course, displaying it for interpretation by the end user. And so if we go to the next slide, you see the combination of all of them put together. So this is being proposed, and the PaLM group is working with DICOM to help better define what is the future digital imaging workflow for pathology and laboratory medicine. In radiology today, there is the concept of a PACS system, which is inherent to the workflow of a radiologist. A pathologist will be leveraging digital pathology images, but they might not use a PACS system. They might use a PACS system, and they might use a PACS system only for storage and continue to use the LIS as the workflow device. And so there's significant thought that needs to go into a new profile that speaks to anatomic pathology workflow encompassing all sorts of images, because although the whole slide image is the future, it is the evolution of our specialty, there are many other images that are part of the pathology specialty that aren't part of radiology, such as the gross macroscopic exam images that are an integral part of rendering diagnoses for patients in the field of anatomic pathology. Next slide. Shifting gears a little bit, as Riki mentioned, this year we have a new profile in the works in transfusion medicine, and specifically in tracking adverse events during administration. So this foray into the world of transfusion medicine is the first for the pathology and laboratory medicine domain, but is hopefully the first of many ongoing efforts so that we can encompass the entire life cycle involved in transfusion medicine, from the point where blood is donated all the way to the end, which is what we're dealing with now, which is a clinical need of being able to track adverse events during administration. So this TMA profile will add workflows and messaging transactions which focus on communicating the administration of and adverse reactions related to blood products from an electronic medical record to an LIS system, incident reporting system, etc. And these additions will update the technical framework volumes 1 and 2. Prior parts of the full clinical transfusion medicine workflow, including patient testing and ordering, as well as subsequent product dispensing, inventory tracking, etc., hopefully will be in additional supplements. And provisionally, we're thinking that these will be, part of a transfusion medication ordering and transfusion medication dispensing profiles to come in the future. We have a couple vendors involved in developing this. We have some subject matter experts, but welcome greater participation in the development of transfusion medicine profiles to allow for interoperability of the various systems for this very important patient care activity.